bioinformatics. So, bachelor at the Department of Chemistry at the University of Patras. Then he did his PhD at the Department of Biology in Athens. And then he moved on to Spain, in Barcelona, and he did his postdoctoral studies. He returned to Greece, and he continued his postdoctoral studies in Fleming and Dimokritos in Athens. And then he was elected as an assistant professor of bioinformatics in 2010. He's president? Yeah, maybe.
Latest 2010.
He joined the department 3 or 4 months ago. So today he's going to present his data. Well, his major interests actually have to do with nucleosome positioning on chromatin, on DNA. Today he's going to present a piece of data in yeast, actually, that have to do with nucleosome mapping upon stress, during stress responses or something. So please.
Okay, thanks, Babis. Thank all of you for being here on such a warm day instead of heading to the beach. As Babis mentioned, I have a sort of wide range of interests. A lot of people have in the past contacted me about data analysis, analysis of next generation sequencing data, with which, of course, I have worked. But my main love, let's say my main passion, has been since relatively early chromatin. So most of my work has had to do with the way DNA sequence may guide chromatin structure in general. And today I would like to talk to you about a very nice, according to my opinion at least, a very nice work that was concentrating on this aspect in a very nice model organism, yeast. So, very brief introduction to things that I'm pretty sure you're all aware of. DNA within the nucleus is not linear. Okay, the analogy of having DNA packed into a eukaryotic nucleus would be like trying to fit a 200-kilometer thread within a nutshell. So this, of course, is not possible unless some very intricate mechanisms come into play that at the same time manage to package the thread, but leave the thread intact in terms of mechanisms that have to do with its replication, its transcription, and its regulation. So, if one looks at the DNA within the nucleus, within the eukaryotic nucleus, you can see various levels of organization. So one can start with its linear view, where you can have genes, intergene spacers, regions that are dense in genes and regions that are gene deserts, regions that are highly transcribed and regions that are not so much transcribed or not transcribed at all. Going on to a second level of three-dimensional organization, you start to see domains forming. And now we know that there are a lot of players coming into the game that modify chromatin, post-translational modification of histones. I'm pretty sure you all know things about that. But there is another level that organizes domains in the three-dimensional space. So the main question is how one goes from the linear DNA sequence into organization of chromatin loops, topologically associated domains, compartments within the nucleus, and finally territories. Okay, territories meaning regions where you have transcription factories, active transcription, attachment to the matrix of the nucleus, and so on. So a very interesting thing is that as you go from the lowest level to the highest level of three-dimensional organization, you gain stability. So territories are stable. It's pretty difficult to push them towards one or another state. But you lose reproducibility. So if you look at one cell next to another cell, you find differences in the way that the topological domains are being organized. But at the bottom of all this, there is one invariant, and that is the DNA sequence. The DNA sequence is invariable of cell type, condition. It's very specific to the organism, and it is conserved. So the main question I'm interested in is, how can one draw information from the primary DNA sequence that would be predictive of these various levels of organization? So how one goes about this, you have to try to restate the question. So how can we link the information from the DNA sequence to the chromatin structure? And for one to try to give an answer to this, it would take, first of all, to focus on a mechanism that affects the organization in three-dimensional space, the chromatin structure. But you would also have to have a readout that could be linked to properties of the sequence. And what can we know of the properties of the sequence? We can study the sequence of the gene, the sequence of the promoter. We have pretty nice tools to give us insight on how the sequence at the local level is being organized. And we need an experimental approach that will relate it to chromatin structure and the DNA sequence. So one nice example of this sort of approach has to do with the way topoisomerase functions. So topoisomerase, for those who are not familiar, I was not familiar at some point, are enzymes that relieve the topological, the torsional stress that is constantly being produced within the cell. One major source of topological stress is DNA replication, but even throughout the life of the cell, topological stress is constantly being produced through the process of transcription. So if one imagines how transcription propagates, what happens is that the RNA polymerase is constantly unwinding DNA in front, and this creates negative supercoiling at the back and positive supercoiling in the front. It's like trying to rip apart an entangled thread. So you can.
Can we interrupt?
Yes, of course, all the time. I have no line of thought, so I will not break.
So I have two very brief questions at that point. First of all, is there a solid validation of involvement of topoisomerase involvement in transcription? First. Second.
Yes.
Second, what happens if O2 complex rotates around DNA? What the need for topoisomerase?
So even if the topoisomerase is moving, even if it's the polymerase that moves or the DNA that moves, because now we know that there is a very great likelihood that this happens, that the polymerase is stable and DNA is
I'm talking about going around, polymerase going around the DNA. So
even in that case, but even in that case, what happens is that there is a twist from the basic form of the DNA helix. It's twisted. So every time you have one twisted, a right will appear. It's pure topology. So it has to happen. It's mathematics.
if the
So,
polymerase goes the helix, then we don't have.
No, but the bubble. This would mean that there is no bubble. You mean that there is no. I think that I'm not very sure about what I'm going to tell you, but I'm more or less convinced that you have to have a bubble. Biophysics tells us that you have to have a region of at least some.
Polymerase is a bubble. It's not longer than 16 centimeters per day.
Yeah, but that's two twists already. So once you do this, and there is data, especially focusing on the ribosomal DNA locus, that show exactly that the process of transcription generates topological stress, and you really need to have both the isomerases to relieve this stress, otherwise the cells go into a state.
There are experiments with plasmids in yeast strains that lack some topoisomerases, and there you find that depending on the orientation of the genes, the superhelicity of the plasmids changes as evidenced for what you said.
What I remember as a solid plasmid was mentioned.
There are some experiments by Jim Wang back in the days, the 80s, where they show this. And it's also part of the.
I think that the most solid information comes from
Yes.
in vitro experiments.
Yeah. Because
That
it is.
transcription changes superhelicity.
Yes.
Transcription, not replication.
The involvement in replication is different. So the absolute requirement of the isolation for replication is to relieve the carbon, right?
Thank you.
Okay. So the idea is then to see what happens if one does not have a topoisomerase in the cell. What happens? And what can this tell us? So our main objective was to link induced topological stress to gene regulation, to answer exactly these sort of questions, to relate the structural features of the DNA to the way the regulation of genes is being organized, and most importantly to investigate if there are different cellular strategies for genome organization. And I'm going to come back to this in the following. So what did we do? We had the yeast strain that was lacking topoisomerase 1 and had a thermosensitive topoisomerase 2. So topoisomerase 1 is not essential, but topo 2 is essential. So what we did is that we used this mutant strain. We deactivated transiently the topo 2 by giving a thermal pulse of 10 minutes. And during this pulse, we gave the cells UTP, radio-labeled UTP, and monitored the output of transcription. So in this way, what you do is actually you see which genes are currently being transcribed or not transcribed during the lack of topoisomerase activity. So you monitor the actual transcription in a sort of a nuclear run-on experiment. So we compared the genes with the use of a microarray to the wild-type strain, and we came up with eventually with very strict criteria around 270 genes whose transcription was increased, they were upregulated, and 160 genes whose transcription was downregulated. But before we went forward to analyzing those, we wanted to clean them up. So we had induced thermal stress in order for the topoisomerase to be deactivated. So we had to remove these genes that were known to be affected by the thermal stress. We ended up with 170 and 93 genes whose nuclear run-on values did not change at all. So this is what you get with the clean data, the downregulated genes before and after cleaning the data, upregulated genes before and after cleaning the data. So this is to show you that the overall effect in the change of the level of transcription was not due to the thermal shock. We just removed the genes that were known to be affected by the heat shock, and this did not change the values. So the idea is now to look at what happens to these genes from every possible angle. First thing that one would obviously do is to look for different enrichments of these genes in specific functions. So what are these genes? Are they essential? Are they usually highly transcribed? Do they contain specific properties in the promoters and so on? Do they belong to specific functions or whatever? So a couple of things popped up immediately, so and more or less expected. The downregulated genes were extremely enriched in essential genes, whereas the upregulated genes, not so much. Actually, they were depleted in essential genes. And one other interesting aspect was that the upregulated genes were enriched in genes that were being regulated by Tata promoters. So they had a clear Tata box in their promoter. We wanted then to dig further into what happens in terms of of functional enrichments, which functions are enriched in each of the two subsets. So if you will allow, there will be a couple of geeky interludes in the talk, so to give you an idea of what is the work being done at the level of bioinformatics. So this is the first one. So in order to exhaustively scan the, let's say, the space of functionality, we wanted to make use of all the data we had, not just the genes that were selected with some threshold criteria. So a good way to do this is by employing a certain approach which has already been published. This is a slight modification. It's called gene set enrichment analysis. This, I'm not going to go into the gory details. It takes all the values within a sample, a gene expression experiment or what have you. And what it does is more or less this thing. So let's say that you have a gene set containing n genes. A gene set can be a certain functionality, a certain area in the nucleus, whatever categorization one may think of. And what you simply do is that you split the number of genes according to how many genes are upregulated or downregulated in the sample, with no threshold whatsoever. You just set the line at the 0. Okay? So this means that m genes had increased values in your gene expression experiment, more than 0, n had less than 0. And what you do then is you just compare this discrepancy with the expected one based on the overall gene expression experiment. Say that you have an experiment that has 20,000 values, 10,000 are up and 10,000 are down. Then the expected would be 0.5. You compare this discrepancy to the expected value, and this is how you calculate an enrichment. And then you assess the statistical significance of this thing with a hypergeometric distribution. You can also do this, and this is the power of this approach, by bootstrapping, because it's a simplified version of the gene set enrichment analysis that allows you to have very fast calculations, so you can also assess the significance more rigorously. In the end, what you have is that you have a great number of values, expression values. You can have any gene grouping you have, gene ontology, pathways, topological domains, whatever, and you can just calculate the direction of enrichment according to one or the other state. So immediately you get functions or pathways that are enriched in upregulation or enriched in downregulation. Geeky interlude over. So we applied this approach to our data, and we came up with also some more or less expected results. So we saw that the upregulated genes are more or less belong to general categories of stress response genes. But very interestingly, we came up with the finding that a lot of the genes are involved in the uptake and the utilization of polyamines. So polyamines are known to somehow assist the cell in conditions of topological stress. They can, let's say, assist the cell to relieve supercoiling by intercalating in the DNA and sort of things like that. The downregulated genes, on the other hand, we found them to be more concerned, so enriched in essential functions and primarily involved in processes that had to do with basic cellular functions like DNA transcription, RNA processing, and protein translation. But this was just the first level approach. The next question is, how are these genes being regulated? So what we did next was to try to see what happens in the promoters of these genes. So what kind of specific characteristics can be attributed to the down versus the upregulated ones. What you see here is that, so these are two identical, two heat maps of identical dimensions. each line represents, each row represents a specific transcription factor. And from red to yellow, you see the enrichment of these transcription factors binding sites across the gene. Okay? So the transcription start site would be here. Bright yellow would mean that this particular transcription factor will have an enriched, a greater probability to have a binding site within the downregulated genes, the promoter of the downregulated genes, and here the same for the upregulated genes. One immediate thing, one immediate observation one can make is that this area is wider than this area. So there is.
What's the size of this line?
This is from minus 500 to plus 500.
Which transcription factors are we talking about?
I cannot discern. All of those are, that are compiled by Harrison et al. In this paper.
By sequence identity, not by real spectrum.
Yes. So these are the bindings. They are quite rigorously defined based on comparative genomics, but they are the bindings. It's not cheap. Okay. So
what is the conclusion from this?
the conclusion for this. So the conclusion for this is that it appears that the upregulated genes have wider promoters. They can accommodate more binding sites, and this is also quantified here. And this is more or less spread out in various categories of transcription factors. But we also employ let's say, the inverse approach. So what you see here is we are now not looking at down on or upregulated genes. We have just the binding site enrichment of each transcription factor for all these genes. What this tells you is that there are transcription factors that have binding which is not very well confining with what we know, like positioning close to the transcription start site or immediately upstream of the transcription start site. And what you see here, orange bars means enriched in downregulated genes, blue bars means enriched in upregulated genes. So one thing that immediately pops up is that for all these transcription factors, which are significantly enriched in downregulated genes, there is a very nice pattern that fits with our traditional view of transcription factor binding, which means a confined area of binding. So again, I have not marked the transcription start site is somewhere here in the middle, and no binding at all within the gene. Okay, so the transcription factor comes and binds right upstream of the transcription start site, and you have no binding, or you have no transcription factor binding sites within the gene. This is not the case for the majority of the transcription factors. And again, we are talking about binding sites. We're not talking about real experiments. But one thing that is showing up is that for these specific traditional-like transcription factors, there is a tendency for these to be found in the downregulated genes. Okay, so the downregulated genes are preferentially bound, or are expected to be preferentially bound by transcription factors that have a confined gene upstream binding. So narrow promoters and no binding within the gene. And also, this more confined binding tells us more or less that there is room for only a couple of transcription factors to bind in these areas. We did this more extensively by employing a very nice computational method developed by an old colleague of mine. So what you see here is this is the area close to the transcription start site, downregulated genes, upregulated genes, enrichment in binding that is now positionally specific. So if you find the binding site in this region and not wherever in the promoter, you get a significant enrichment. What you see is that for the downregulated genes, you have only a couple of transcription factors having a positional enrichment, and this is confined to a small area close to the transcription start site. Whereas for the upregulated genes, you have a lot of binding, a lot of transcription factors, extended areas of binding both upstream and downstream. And what we did next is that we took So this implies that there is a sort of, let's say, more strict regulation for the down-regulated genes, and a more versatile one for the up-regulated genes. At least this is how we interpreted our data. And we wanted to see whether this could be reflected on the way genes are actually being expressed. What we did then was to get data, expression data from various conditions, and there is a very nice public data set of those, where you have gene expression for yeast in all possible conditions that they could get their hands on. Yes, Tash?
Can I ask something? So you're interpreting this view statically, right? But this is a time average in that sense. So instead of this wideness, it could mean that you have somehow like a very fast exchange of transcription factors in these genes that actually shows a wider area, not that you have many transcription factors across this area.
Exactly.
Is that a possibility?
Yeah. So you cannot rule out, and this is the more likely explanation. You're saying that we're not having all of those at the same time. Obviously, no, you're not. It means that they could be accommodated. That given a certain condition under a certain, let's say, layout, you can have this binding, this transcription factor binding here, or here, or here, or another one here. Not all of them at the same time.
I'm losing you. Is this a theoretical prediction for these
Yes.
binding sites or real binding?
No.
So how you would explain the theoretical existence of the binding site with the kinetics of on and off for a given binding factor in the average experiment you're doing?
Now I lost you. No.
How you would explain? I'm coming back to the previous question.
No, but you cannot explain it. You are not trying to explain it. What this tells you is that there is room for a lot of binding factors in these genes, but not in these genes. So you don't expect a huge collection of binding of transcription factors to come and bind wherever in these genes promoters. You can only have a few.
I have to
and they will
remind
be
you the reality from chip-to-chip experiments, which is that it's a very rare event to find the real binding downstream of the transcription steps.
yes, but we have not tried to go into the reality of chip data because it's tricky, slippery territory. We're just looking at what the sequence can tell us. But what expression data do tell us is that if you look at these expression profiles over a great number of conditions, and you just say, okay, for each gene, I want to know which is the highest and the lowest area of expression where it can reach, you see that there is a significantly greater expression variability for the upregulated genes as opposed to the downregulated genes. So the downregulated by Topological genes have a very confined promoter architecture and have an expression rate that is very robust compared to the upregulated genes. So this can vary, and this fits well with stress response enrichments and all this.
Does this also fit with the essential and non-essential genes, or it's just?
It fits with most of the things that we have seen up to now. So essentiality, conservation, constitutive genes are enriched in downregulated. Yeah. So basic functions, they are robust, they don't change, they are not subject to different transcription factors, and they have what the new thing here, the newest thing here is the size of the promoter and the architecture of the promoter.
Makes sense.
So this is where nucleosomes come into play, because we know now that nucleosomes and transcription factors compete for binding, especially around the promoter region, and a very nice predictive index for transcription factor binding is the existence of a nucleosome-free region immediately upstream of the transcription start site. So what you see here is an average of overall yeast genes, the green line, of nucleosome binding around the transcription start site. So you see the nucleosome-free region, a deep cleft in the nucleosomal binding profile, and more or less relative spacing of nucleosomes once you go into the gene. So coming up is the second geeky interlude. What you also see here is an in vitro approach. I'm not going to talk about this. And a theoretical model for the prediction of nucleosome position. So there has been a lot of work in this area before the advent of ChIP-seq that practically solves the problem, because now you can run an experiment and find the nucleosomes. But back in the days, I was trying to find a way of predicting nucleosomes solely from the DNA sequence. And this may come handy for at some level, when you're just interested in what the sequence may tell you, regardless of conditions or cell type. So the idea was to find a way to go from the DNA sequence to the nucleosome position, predicting whether a specific area of DNA will have increased or decreased probability to bind the nucleosome. And our model was based on a purely ab initio hypothesis that had to do with the structure of the nucleosome. So this is the golden and green is the DNA wrapped around the histone core, the octamer. What we know from crystallography is that the lobes of the nucleosome are bent. So the DNA, it has increased bending in order to spool around the octamer. But this is somehow relaxed around the diad axis. The diad axis is the area where the DNA crosses the double axis of symmetry of the octamer. So the idea was that if one would look at the predicted tendency of DNA to curve, to bend, you would expect that the perfect nucleosome would have a shape like this. So bending would increase and the DNA would try to go into the nucleosome, slightly relax around the diad axis, and then have a symmetric pattern as it goes up. So a symmetric DNA curvature with a local minimum in the middle. So this is what we expected, and this is what you get if you look at the actual nucleosomal sequences based on a theoretical prediction of DNA curvature. So our approach had to do with transforming the DNA curvature, so the theoretical calculations of DNA curvature, pass a certain filter, you locate the local minima, and then you ask for these minima to be in the middle of symmetrical curvature. So this is what these equations tell us. So you go from a curvature profile that is not so informative to a symmetrical curvature profile that is more informative. And these are the actual nucleosome positions of the first sequence that we looked at. This is the mouse mammary tumor virus sequence from Otter.
Sorry, why did you calculate the DNA curvature?
So there are theoretical calculations that have been introduced by Chris Hunter and Drew. So these are papers published back in the late 80s. So we know that we know of ways to calculate the bendability. They are all based in. I'm not going to go to very much detail. So if you look at the DNA, you can look at it as a ladder where each base pair forms a plane. But each base pair, compared to the previous and the one after that, are not identical. So there are chemical and stereochemical constraints that tell you whether they tend to tilt or twist more or open like this. So if you have a way to theoretically calculate these three angles, these 3 degrees of freedom, you can have something that additively will bend the helix or will leave it rigid and so on. So this is what. So it's based on experimental data from crystal structures of DNA, small crystal structures, Upon this, you build the prediction for the curvature, and upon the curvature profile, you build the symmetric factor. So to cut a long story short, this is what our model looks like. For the light gray line is all the yeast genes. It's not very well discernible. But what we're really interested in is the red, which is the downregulated genes, and the blue, which is the upregulated genes. Two things pop up. There is a narrower nucleosome-free region for the downregulated genes, even compared to the average of the yeast genes, and especially compared to the upregulated genes. So you see that there is even an area where an additional nucleosome could be accommodated in the same area that the upregulated genes don't have a nucleosome, are not predicted to have a nucleosome. And another thing is that once you go into the gene from here on, you have a more regularly spaced nucleosome array in the case of the downregulated genes, but not in the upregulated genes. And if one tries to model the, let's say, preferential positions of nucleosomes for the downregulated genes, they will be like this. And you see how the cleft, the nucleosome-free region, is closely related to the narrower promoter we saw for these are the binding sites we saw earlier. And it's a different case altogether for the upregulated genes. A wider promoter, a wider nucleosome-free region, and a wider area of possible binding of transcription factors. We also had the chance to test some real experimental data on nucleosomes, and not any nucleosomes, but nucleosomes that are termed to be fragile nucleosomes. These are nucleosomes that, upon increased time of digestion with micrococcal nuclease, tend to vanish in the promoter. So the traditional way of mapping nucleosomes in an experiment is to digest with micrococcal nuclease, extract the fragments, and sequence. But depending on the time that you do the digestion, you get more or less fragments. You can see nucleosomes that pop up in your experiment if your time of digestion is shorter. And these are the fragile nucleosomes that eventually leave the promoter if your digestion is long enough, but are still there if you apply shorter times. When you look at the fragile nucleosomes, first of all, you see them preferentially in the gene upstream regions, in all cases. But what happens is that they have an almost twofold enrichment for upregulated genes. So this is upregulated genes, downregulated genes, and all genes in light gray. So it's not that this area is nucleosome free, but it's that there are probably nucleosomes there that are easier to kick out. And if they are easier to kick out, then probably this gene will be transcribed more easily, even in the presence of a topological stress, because the dynamics of the sequence tell the gene to be prone for transcription, even if chromatin is entangled. Or that's at least a simple way to interpret this.
Okay, this is understood because these are stress response genes, right? Well, therefore they have to be poised, they have to respond fast, and so on. But this still does not explain mechanistically how Topo selects in a different way one or the other set of genes.
How Topo selects?
Selects for this effect in these two different gene categories.
Yeah, so
we don't know that. So we don't know, for example, where topoisomerase binds, or how noxious a DNA break is due to the lack or the excess presence of topoisomerase.
So we know, we have an idea. There was a PNAS paper, I think 2 years ago, that showed an increased binding of Topo2 near the promoters of constitutive genes. But this is quite an obscure article. We weren't able to extensively replicate the data, but there are hints towards that.
I think the major problem for this experiment is that, I'm coming back to my first question, is that always the problem is if topoisomerase, whenever it's detected, And in transcription, I don't think that has ever been detected. But always the problem, if the topoisomerase detected, is involved in replication or transcription. And probably this is something we have discussed about your approach, because in this case, using temperature-sensitive topoisomerases plus a defective topoisomerase.
No.
The first thing you do in a fraction of your cells is a replication.
So no, but for topo 1 at least,
There is no phenotype, I know that.
No, but when we know that it plays a role in transcription, you cannot have transcription in certain genes if topo 1 is absent, especially the ribosomal DNA loci. There's the problem of relieving the negative supercoiling at the back of the polymerase becomes too big because these are very actively transcribed genes, and you.
Even in this case, your topo effective strain is also replication effective. No?
Yes, but you don't
What
allow.
we get here?
No, but you don't allow this to come into the game because you only deactivate it for 15 minutes. The cells do not need to replicate in the absence of
A fraction of your cells, maybe more than 25% of your population, is in a space.
I see what you're saying. I'm not sure if these cells were synchronized, but even in this case,
only if the cells were synchronized, then function would be.
We're entering territory that is not. But even in this case, I think that if there is an effect, you can see it. We have evidence from many areas that tell us that there is a role in transcription. Anyway, but
I hate to disturb the line of thought.
No, there is no line.
That's what I'm saying. But the question is, have these experiments been done with topo 1, with a topo 1 mutant, or with an inhibitor of topo 1? Or have you repeated these experiments not with a temperature-sensitive mutant of topo 2, but with drug-inhibited topo 2, like a topocyte, enicocyte, or camptothecin inhibited topo 1?
We haven't done this, but I think others have done it. Not genome-wide, but for specific gene loci, and they see that there is an effect in transcription.
So, but topo 1 was not tested at all in this approach.
No.
And what was the
So
reason
topo
for
1 is
saying-
absent in this approach because we only want to see what happens with topo 2.
No topo 1 mutant.
Topo 1 is knocked out in this.
Topo 1 is missing anyway.
Yeah. So we only see the effect. We're trying to see the effect of topo 2 alone. The main problem of this is why one should have upregulated genes. So the downregulated genes you can easily explain, at least the way I see it.
But how do that assessment fair? Because this is actually an important point. The way this experiment is done introduces some ways to effect changes that are not directly related to a topo stress. For example, heat shock genes or other stress responses might be activated and contribute to the upregulation or downregulation of genes that you see. So this could not only be attributed to the topo stress, but also to other stresses that have to do with the experimental approach.
So we try to control for the heat shock. It's what I showed in the beginning. So we try to remove as many heat shock so that there is a.
But other stress responses are also not just.
Other stress responses. They are probably going to be indirectly linked to the only stress that we are reducing. And the only stress we are reducing is the topological. If this has secondary stress effects, I'm not really sure. I cannot say.
I mean physiological changes, like for example, uptake of nutrients or other metabolic changes.
Yeah, but all these things we are trying to control with a very limited period of deactivation, because we're actually measuring a pulse in transcription. We don't allow the system to be under stress for a prolonged period of time. So we are thinking that. Sorry.
That's for that 15 minutes.
The run-on experiment.
The 10 minutes, 15 minutes.
So the run-on is 5 minutes, and the total shock is 15. So it's 10. Then at minute 10 we add the labeled UTP, and then after 5 minutes we take the the measurement.
You don't know if this is reversible. I mean, when this 15 minutes pass and then the temperature goes down to the normal level.
So that's how you normalize. You normalize against values that you take afterwards.
So,
This is how a random is done. So you take the measurements at the peak, and then you allow for a quenching time, and then you repeat the measurement, and this is what you normalize. This is your background.
so DNA repair genes, in which category are they?
Sorry?
DNA repair genes, in which category do they belong? At segregate.
Do you see DNA repair genes in the up or down regulated?
I haven't. This is something that hasn't popped up, so I don't know. I would say no, or at least not significantly. This is a small number of genes, so okay. So overall, coming back to the nucleosomes, what we see for the nucleosome binding between the two genes, I'm gonna go a bit faster now, is that overall there are deeper nucleosome-free regions, both upstream and downstream of the genes. This is also something that we know of, so for overall nucleosome maps. And again, a denser population of the body of the gene by nucleosomes. And this is also shown here with real data, so experimentally positioned nucleosomes, where you see that this is the average spacing of nucleosomes. The spacing is significantly higher in the upregulated genes. So you tend to have more sparse nucleosomes in the body of upregulated genes against downregulated genes. I'm gonna go through this, and I will come back to. I will come now to what, to me at least, is the most interesting thing of this work, and it has to do with the localization of the genes in the chromosomes. So we have downregulated genes, we have upregulated genes, but where are they in the genome? So what you see here is a map of the yeast genome. Blue lines are upregulated genes, red lines are downregulated genes. You cannot immediately say that there is a non-random distribution, but you can trust me that it is non-random. They tend to cluster. So if one looks at NRO values that are significantly high for a run of genes, these are found much more frequently than expected by a random approach. So genes that are downregulated or upregulated tend to come together. They are not points in the map. And if you apply this a sort of clustering of genes with similar values that come in arrays of more than 10 genes, you find clusters of these genes, and you see that they are not randomly located. A lot of upregulated gene clusters are located in the long arm of chromosome 12. We'll come back to this. And also, if you just look at the genes compared to some specific marks, let's say of the yeast genome, you find that they are also non-randomly distributed there. So this tells us the percentage of genes as a function of the distance from the telomere. This is the average, so more or less around the average gene frequency. Downregulated genes, upregulated genes. Upregulated genes tend to locate close to the telomeres. Downregulated genes tend to occur close to the centromeres, away from the edges of the chromosome. And this is statistically significant, and there is also a disparity in which of the two telomeres the upregulated genes are localized. So within any given chromosome, they will tend to be located as one or the other end of the chromosome. More importantly, if one looks now at the clusters, what happens to the clusters? Here, what we see is that the size of the spacer between consecutive genes that belong to down or upregulated clusters. And what you immediately see is that upregulated genes have wider spacers in between them. If you look at a cluster of upregulated genes, the spacing between the genes would be wider compared to the space between a cluster of downregulated genes, and this is also true for both upstream of the transcription start site and downstream of the transcription start site. So what you see here is that there are sort of neighborhoods that are defined at the level of the response to topological stress that have specific, let's say, geometrical properties. Okay? And one other interesting thing that sort of, I think, sums up the picture is the direction of the genes, which was mentioned before. This is very important. So if you look at what this is almost probably the most complicated slide, I'll try to explain this. What we have here is the genes centered around the gene that has been affected by the approach. Red is downregulated, blue is upregulated, and what we plot around this central zero is an arbitrary value of the direction of the genes being transcribed. So these are 5 genes upstream and 5 genes downstream of a central gene that has been affected. What we see, what this tells us is that genes that are being downregulated are positioned in areas where you have alternating transcription direction. So you have something that looks like this. Whereas around upregulated genes, you have collinear transcription, so co-directional transcription. The genes are either all this way or all this way. The value here is arbitrary, and this is true also for the complete data set. It's not only this is based on our genes, the 170 and 90 genes that are being affected, but if one looks at all the genes that are either located in the middle of genes being transcribed like this and like this, are genes, say, caught in the middle of opposing transcriptional directions, you overall have lower nuclear run-on values. Whereas if you pick genes that are in the middle of a co-directional transcription, you have increased nuclear run-on values. So,
are the genes co-regulated physiologically?
I have not looked this. We have not seen this. It's an interesting point. So what this tells us is that you can think of 2 genomes. What I like to call the suburban genome is located at the telomeres. It has long spaces in between. Promoters that are wide can accommodate a large number of transcription factors. You don't care about the nucleosomes that much. They can be more sparse within the gene body. And they are very, very nicely lined up with streets. The genes are one next to the other. And importantly, if we assume that there is a topological stress induced by transcription, this is what it will look like. Negative supercoiling, positive supercoiling. Negative supercoiling, positive supercoiling. Because the direction of the transcription tells us this. Whereas there is the old city center with all the conserved functions, the essential genes located in the middle of the chromosomes towards the centromeres, and these tend to be crammed up. They have smaller space in between them. They have to have a very strict regulation because there is no room for transcription factors to bind, and also there is actually no layout. You have genes like this, like this. The direction is not, let's say, laid out in a way that allows for the dissipation of supercoiling. This is what I'm trying to get to. So a region like this will tend to be less dependent on the action of topoisomerases, because the supercoils will tend to cancel each other out, and there's also greater spacing between the genes for the DNA to relieve the tension. Whereas if you have something like this, with genes very close to each other, and with the direction of transcription imposing accumulation of supercoiling, you definitely need a protein to relieve the stress. So, last thing, I'm going to touch a bit upon the 3-dimensional structure. We saw that there are clusters of genes that are dependent on the effect of the topoisomerases, but now we also have 3-dimensional maps of the yeast genome, with which we can compare. And what we immediately see is that there is the most significantly enriched area in upregulated genes is the long arm of chromosome 12, which you see here in a 4C map. So this is the whole chromosome 12. This is the centromere. This is the ribosomal DNA locus. And you see that the chromosome is actually split in two. There are a lot of interactions in this part, but they are not connected with each other. This shows up in a 3D reconstruction here. So here, this is an area where you have a lot of upregulated genes. And you see that it is very distant from the core of the nucleus of the genome where all the centromeres cluster together. And also, this is a reconstruction of the map we saw previous. This was very recently published. That tries to define specific territories, communities within the genome that are said to be communities by an increased degree of interaction between them. So there is one great community that is called the centromeric community. It's formed by mostly the centromeres of all chromosomes and a large part of chromosome 1. And this is where you have the most increased enrichment of downregulated genes. So we knew this already, but now we see it in the three-dimensional space also. Whereas there are areas, with the most particular one being the chromosome 12, downstream of the RDNA locus, where you have the enrichment of the upregulated genes. So to sum things up, what this approach could tell us is that, first of all, there are functions that are related to basic molecular processes that are mostly affected by topological stress. And this is something that one would expect. There are genes that have a confined regulation pattern. They have smaller promoters, very well positioned nucleosomes, and these are the ones that are more dependent on the effect of topological supercoiling. Last but not least, there is a very clear tendency of the relative organization of genes in linear space. So the way genes are put next to each other may have a very severe effect on the way the cell, the nucleus, can relieve topological stress. So you have various levels of organization, and this, of course, can also be related to the position of these neighborhoods in the three-dimensional space. So overall, you have conserved essential functions being buried in the core of the nucleus around the centromeres, and these genes are smaller, tighter together, and with no specific motif of directionality, whereas you have located in the suburbs of the genome genes that allow, let's say, a more versatile response to at least topological stress. This may also apply for other stress, because most of the stress responsive genes that we look are located in the outer areas of the genome. So that was it. This work was pioneered by Joaquim Roca at the IMBB in Barcelona. This was the one who, Joaquim was the one who convinced me to look into the toposomerasis. All the experiments were part of Nacho's PhD thesis. He's a very nice guy. He's now retired from science. He's into the music business. From time to time we talk about Barcelona beating Panathinaikos in basketball. And the link between these two was my former supervisor, Roderick. He largely contributed in all the work that had to do with the nucleosome position. Panagiotis Kouvardas and George Kollias, under whom with George we supervised Panagiotis' thesis, is the responsible for the gene regulation, the functional gene enrichment tool that is now being prepared. Thank you. I hope I was not too long. And I don't know if there is room for And now we can discuss if toposomerasis affects.
No, we discussed that already. Let's discuss something else.
So what about, for example, if you want to validate some of this data, what would you do?
tricky. First of all,
individual genes or promoters.
I'm not sure I can really answer this. My view is always the big picture. So first of all, I would try to do a chip on Topo2. But this can also be tricky, as I told you before. There's already something published. Validating this data, so one thing that would come to my mind would be to construct an array of co-directionally transcribed genes and see whether I see different things in an in vitro system. So an array of 5 genes. I'm not really sure how feasible this is. Nothing.
Put them in vitro in a transgenic system. So put it in the yeast and see how they work, or if they're affected by topo.
But then the problem is that you're not really sure of where this thing would be located in your nucleus. So if you have,
I guess you can find it.
Yeah, but I mean you cannot rule out that the effect will be an effect of the general environment. Because if your cassette, let's say, gets to be buried in the core of the nucleus, then you may be seeing something that is not dictated by the relative position.
So see what imposes to what.
Okay, yeah, but this is a different level of approach. Then you can also do this and like locate it with fish or some other technique.
Not the level system, but if one looks, for example, with antibodies against topoisomerase, or do chip experiments, you see a specific area that topoisomerase is enriched in.
Yeah, as I told you, so we know that Topo2 tends to be enriched in constitutive genes promoters, which is, this fits well with what we see. The main question has always been, why should one have upregulated genes? And a lot of upregulated genes, because they're almost double the ones that are being downregulated. And even after having ruled out heat shock responses to a large extent. Why should one have...
Why do you think upregulation is not is an unexpected response?
Because the idea is that topological stress should equally affect all sorts of genes. But what these, especially the last part of the results tell us, is that there are some that may even not need it. If this is true, if the co-directional hypothesis of dissipating the supercoiling without the need of an external force is true, then it means that these genes do not care about topoisomerase. And if they are highly transcribed, they would even need it less, because the supercoiling would act as a pump.
Yeah. But this is mainly assuming that all the regulatory regions will have a positive effect on the genes, which is, I think the data also supports that this is not true. Maybe the negative effects on regulatory regions are more important. So the tension you apply removes them, and you have more regulation.
I didn't get it. What do you mean the regulatory effects?
The regulatory regions. So they may not have only a positive effect on the gene. They may have
There may be suppressors.
But actually, part of this problem is that you don't know how many of those are direct targets, and not secondary targets.
Yes, you don't.
We have done. It's similar to that.
My interpretation of what's happening, and based on Topo1 and Topo2 mutants in yeast, it seems that we have a system where, Topo1 is essential for relieving the torsion stress that's associated with transcription made. However, in a Topo1 negative background, Topo2 can rescue the phenotype. Okay?
Yes, because
and cells will be viable.
If
this based on the mechanism of Topo1 and Topo2, Topo1 has only one of the two strands, so the action is local. Topo2, in complementing the phenotype, will have to act at a distance. So, because it has to pass two pieces of DNA one through the other, right? So, a double strand break and then passes over the double strand. So, the action is indirect. I do not expect to see Topo2 binding in the locus. What you're seeing is an effect between regions of the overall nucleus that need or require fast disentanglement or like release of stress, and other regions that do not need this immediate release of stress in this time frame.
Yeah.
So it's an indirect effect. I think the whole discussion about where Topo 2 is located is irrelevant.
So
if
what
you're using this as a tool,
so
yeah, so
yeah.
it's relevant, right? You're using this as a tool
It's not relevant, though.
why relevant to
Because
what you
it's
see.
propagated.
Yeah,
It's not easily interpreted.
I'm saying the only person that probably is not there. If we would not find it. So it's probably acting as a distance.
I totally agree with you, and I have to add, if you allow me,
Yeah.
that if this was the case, then for each round of transcription, that means for one or two polymerases, scanning the whole volume sequence, then you would need a topoisomerase function.
You need it. So this
For
is
each round
no,
of
no.
transcription.
What happens
So, you see what I mean?
yeah, but it's there.
So for an actively transcribed gene, for a couple of rounds, you would need a couple of strands DNA break, so on and so on. This has not been described.
I don't think that's
DNA damage and repair happens once, but not as frequent as transcription.
But I'm not entirely sure, but I think that biophysical studies, have shown that the level of ATP needed for transcription cannot be explained simply by the needs of the polymerase.
You need one molecule of ATP for each nucleotide that you incorporate. So there's a huge amount.
So it takes more because you need the topoisomerase 2 to relieve the positive supercoiling. So what happens is that Topo 1 is not essential. Topo 1 relieves the negative supercoiling downstream of the transcription start site. But the great majority of DNA is negatively supercoiled. So it's not a big burden for the cell. What is a big burden for the cell is the positive supercoiling ahead of the polymerase. And there you need the Topo 2. And this is why Topo 2 is preferentially bound, let's say stationed, to the genes that will have a large round, many rounds of transcription. Because the positive supercoiling there will be so much that it will bring the cell to a state. This is how I'm interpreting this. But I'm pretty sure that there are data showing for specific loci that you need Topo 2 to have transcription, at least propagating transcription.
Would you expect in these different neighborhoods that you described that you have different chromatin modifications, and that they playing a role defining these regions, or could they be a consequence of the existence of these regions?
One, yes, I would, and two, I would bet for the second, so that they are the modifications that we would have there would be more an effect of the.
But it should be possible to find that out, or not?
It should be. I don't know how much work has been done for this in yeast, but for instance, if one would do the same in cases for which we have a lot of, because they are very much linked to specific conditions and cell types, at least in higher eukaryotes. You have chromatin states dependent on post-translational modifications that are cell type specific. So I'm not very well informed on yeast on this aspect.
This structure which you described, the essential genes around the centromers and the up and down regulated genes at the edges, does this hold also for multicellular organisms?
I cannot say. And it's only the upregulated genes at the edges. The upregulated genes by topo stress.
Another question. You tested one trigger. This was some kind of stress. If you change the trigger, what happened to these genes?
We didn't, and
which are lined, are in one direction.
Yeah, I know. We haven't, because my guess would be that it will not be the same. There will not be an effect. If our hypothesis on these genes being like this, in order to not be dependent on topological stress is correct, then you would only expect those to be affected by topological stress, not other sort of stresses. But it's an interesting point. I don't know. We were interested in the topology.
Shouldn't topological stress be relevant for a linear chromosome if it's allowed to twist like that? Unless if its end is not anchored someplace. Should topological stress be relievable without any topoisomerase just by twirling the chromosome?
Yes, it is, but it's impossible to happen because of the length. So with the length of any chromosome,
But also these are in many positions, so it's also anchored. It's not free.
Yeah, there are anchors.
Not
There are anchors.
That's right.
At
It's
various
anchored
points.
too much.
Yes, too much.
So if you would have a 2,000 base pair chromosome, yeah, probably, but
So if what you are saying is true, then using mammalian chromosomes, the effect is going to be even more severe.
Because they're more anchored.
the greater the complexity of the territories, the greater the severity, yes.
But then that's why maybe genes are more suburban when you move to bigger.
Yeah,
bigger
that's an interesting. I've thought about this. It's an interesting.
They don't cluster too much.
Yeah.
From the evolutionary point of view, what is the relation? Is there any relation between the suburbs and
Yeah.
cities?
So the city center is conserved.
Is it?
The suburbs are the new city dwellers.
So your analogy is perfect.
It's good, I think.
Thank you.
Thank you.